But welcome to Neuroccino. Sorry that it was a bit hectic now. There is no live stream today, as you just realized, because we got locked out of the account. But we have 3, if not more, fantastic papers this morning. And I'm going to hand over to Victor first, who has a very apt paper for a Monday morning. Take it away, Victor.
Hey, thank you. So the paper was published in Communications Biology a few days ago, I think, and it's about brain size and neuron numbers drive differences in yawn duration across mammal and birds. So you may have seen on social media like Twitter a little spoiler from this article about yawn, and it's here. It's this figure here. It's because it's trying to, it's a paper that is trying to solve, or at least that gives indications to solve the mystery of why are we yawning? What purpose does it serve? And so it explains that there have been different hypotheses. For example, one that was saying that it's to regulate the oxygen level in the blood because we are breathing like a big gulp of air, But this one has been, this hypothesis with the oxygen has been falsified over 30 years ago. Now the most the hypothesis that is the strongest at the moment is that it is a mechanism that helps brain cooling, and so there have been different experiments that have been done to test this this hypothesis, and it seems that the temperature of the brain and of the body, but also of the brain, is linked to when we yawn, and the fact that when we yawn, it reduces the temperature of the blood in the brain. So it seems to go in that direction. And so what they propose is that the bigger in this article is that the bigger the brain and the more neurons you have, the more heat you will generate inside of the brain. And so you might need more of a thermolytic effect of some kind of behavior to compensate this big heat generation in the brain from time to time. And so this would mean that the bigger the brain, the more neurons you have, the longer your yawn would be. And so this is the hypothesis that they are testing it here. So they wanted to try to test this hypothesis on as many animals as they could, so that they have a very large sample size of different species. And they do it both in mammalian species and avian species, so mammals and birds. And so they tested it on 1,291 yawns from 426 different mammal species, and 271 birds across 101 different species, which is like, when I read that, I was really astonished, because it's a very big number, and big respect for this kind of research, because it must take quite a lot of time and dedication. They could not. They tested also the brain size and the neural count, but the neural count, they did not have data for all the different species that they had. So it was on a subset of these species. To test their hypothesis, they used a model called Bayesian multilevel phylogenetic models to relate the brain size and other brain measures with the yawn duration. these models, I'm not super familiar with them, but they are models that take into account the relationship between the different animals in the phylogenetic tree to say, okay, these animals are close together, so they are not independent, their measures are not independent, and it takes that into account when calculating the significance level and stuff like that. So I'm not really, I don't know much about it, but it's a whole field. It's quite interesting to know that there is this kind of thing. So they measure yawn by the moments of the closed mouth to the closed mouth. So you have like an example here of a mongoose, and there of an emu, and and a few examples of different yawns in different species. And I hope it's not making you yawn too much on this Monday morning. Here is a figure of, I think it's a nice figure, but not extremely informative. The more informative will be later, but it's a nice figure showing the standardized trait value, the trait being either the yawn duration or the brain mass here for the mammal and the birds. So it's the color that is encoding the trait value, and it is put on the phylogenetic tree of each clade. And so you can see quite a nice parallel between the brain mass and the yawn duration colors here, and also on the mammal tree, in the mammal tree and on the bird tree. The length of the bar is the phylogenetic differences in million years, I think. And here you have like the more precise measures with the predicted association between the different brain measures and the yawn duration across different taxa. So on the top row, it's for the mammals, and on the bottom row, it's for the birds. So you've got the standardized brain size, brain mass, neural count, and cortical neuron count for the two different type of taxa. And you can see a very nice correlation between the two, between each brain measures and the yawn duration. So the bigger the brain, or the more the more the neuron, I don't know how to say that in English. The more the neurons, okay. The longer the yawn, and same for the standardized cortical neuron count. So total neuron or cortical neuron, same. This relationship is also there for birds, but not as strong, but still very much there. So very, very good. Then what they did is to control for the body size, so that they did not take into account the body size. Well, I think they regressed out the body size in their model to only get the residuals for the brain mass and the total neuron count, and they still get, albeit smaller, but they still get a positive relationship between the two measures. But to note here, what they are measuring is not really the brain mass or the neural count. It's more the difference between the expected brain mass, for example, at a specific size, and the brain mass of this specific animal. So it's a bit different than what we had above, but still we have a positive relationship, which is supporting the hypothesis. And lastly, the last measure that they did was to model, to take the average brain mass of a bird and to check what would be the yawn duration for this brain mass to pair this brain mass to mammals, saying like a mammal that has the same brain mass as the average bird brain, what would be the yawn duration? And what you can see is that the bird yawn duration is systematically and very significantly shorter than the one of an equivalent mammal. And it's true also for the body mass, when you take the body mass and the yawn. So this shows that birds have a shorter yawn than the mammals, even if you take into account the body size and everything. All in all, this nice paper demonstrates that there is a very strong relationship between the brain size and the duration of the yawn, and that this goes perfectly into the hypothesis that this brain size is that the yawn duration has evolved to be used as a thermolytic behavior to control the brain, the temperature of the brain. And the difference that they observe between the bird and the mammals can be explained in different ways. First, there is the fact that birds have a temperature of their blood that is in average about 2 degrees Celsius higher than the one of mammals, meaning that there is a bigger gradient of temperature between the blood and the air, meaning that the heat will go faster from the blood to the air in the birds. So they don't need as long as a yawn to remove a specific quantity of heat, for example. And that could explain the difference that we observe. Also, they have what is called where is it? I can't find it. Well, they have a structure in their cranium that helps the dissipating of the heat. So that could also impact this kind of thing. They also have a beak that can be used for radiative cooling. So that also has to be taken into account. And so there is a lot of different things that might play for this and explain this difference. And so it's still quite intricate, but it's a big step forward in this field. And just to finish, they also talk about one big outlier in their mammalians, which is the naked mole rat. I don't know if you know this little guy. It's like it's a rat that doesn't have any hair and live in colony under the underground. And these rats, they have a very long yawn for the size. They say that they have the same length of yawn than the jaguar, even though their brain is like 300 times smaller. And this was quite strange. But one other thing is that it's a unique thing about this naked mole rat is that it is poikilothermic, which is the same kind of thing that we have with lizards or stuff like that. They regulate their temperature with the environment. So this is something that is quite unique for them. And this might explain some of this weirdness that they have. And so that would be also very interesting to check and extend the current line of research by checking the ectothermic clades like saurians and other lizards, because we know that they also yawn, but this study did not include them. And so this would be interesting to see what's happening with that. And it's I think it would be really a nice way to continue this kind of research. But yeah, that's it. If you have any question.
People kept writing me in between. Super exciting study, super interesting for Neuroccino on Monday morning in particular. Right, Patrick, you were the fastest. Go.
So this is obviously a fun paper. I've got so many issues or so many questions. First of all, has it been shown that yawning actually affects the heat development in the brain? Like, has there been any study with a thermo measurement of the brain and indicating that indeed, if I yawn, my brain gets cooler? Because like my mouth isn't really a device that cools my brain, isn't it?
Yeah, so what they say about that is that yawns are triggered during rise in temperature and followed by corresponding decrease in temperature. So this is what they say in the introduction. I didn't check the references. I just looked at the paper, but this is definitely something that they say has been checked and is one of the reasons this hypothesis is so strongly defended today.
Sure. I mean, I don't debate that it might not cool me for some reason, but does it have an effect on the brain? Next up, regarding their statistics, if the hypothesis is true, so if this is the function of yawning, then we should see differences in yawn duration within species too, because different members of one species have obviously still different brain sizes. So if it's really neural count or brain size, it should still be possible to find a correlation between yawn size or yawn duration and then brain size measures.
Not necessarily, I think, because if it has evolved as like a general mechanism and not something that is tightly related to thermal indicator in the brain or anything, it might be something that is automatic and which might provide like a little help, but would not necessarily be completely tied and well related to the direct temperature, I think. But I don't know exactly. I think it's a good remark, and maybe it can be checked, but I don't know how exactly they could do. Well, I mean, I know how they could do it, but I don't know if they checked it. And so, yeah.
Well,
good point.
Probably it would have been good for the human connectome data set to also measure yawn duration of the human participants. Probably that would be the argument here. Anyway, really fun paper. Thanks for presenting.
Thank you.
There was a question in the chat as well, whether they measured correlations between oxygen intake and yawning.
No, I don't think. No, not at all. Well, I didn't see it, but maybe somewhere it's buried in the article, but I didn't find it. So it was really about the just the yawn duration as seen, well, visually inspected. I think. I don't think they checked for oxygen or anything like that. That would have required quite a different setup, I think, because it's a difference between a camera and an oxygen apparatus, of measuring apparatus. I don't know.
Okay, sorry, I was asking that. I tried to speak because when I was like reading something about the brain and jawing, I have this memory that you do this for like increase oxygen when you're tired, maybe, but maybe I'm not if I'm correct. So I was thinking about that just because
well, that's
one thing that I know between the brain and jawing. Yes,
that's one thing that they first talk about in the beginning of the introduction. They say like, yeah, this hypothesis about oxygenation, yeah, it's been falsified 30 years ago and still being talked about. And they say, I don't remember exactly what they say, but yeah, that, for example, it's been demonstrated that altered level of oxygen and carbon dioxide do not alter yawning, and physical exercise sufficient to double breathing rates has no effect on yawning. So yeah, doesn't seem that the oxygen level has any relationship with yawning.
Okay, and I was asking because it what I heard like years ago, but as you say, it's difficult also to evaluate that measure. Yes, true.
But yeah, I'm with you, Leah. I also had that oxygen story in my mind. And when Victor presented the paper to me, I'm like, cooling the brain? I never heard of that.
Yeah, and that's exactly why I think they put it like at the very top of the articles, like it is not oxygen.
Sorry, since I was looking for that.
No.
That's the whole point of doing Neuroccino, right? To learn. But I'm glad I'm not the only one who made the mistake.
Other questions?
I'm wondering if we have genes for yawning. Why we keep on doing that if it's not effective?
I mean, maybe it is. They're saying it's good for cooling the brain, so maybe it's not useful and it's just something that stay there. But they say that it's weird that we would keep it even though it's useless, because there is a risk with yawning of subluxation of the jaw and locking of the lower jaw that can happen. Yeah, and so this is selective pressure against yawning, and yet we keep it. So there must be some advantages. So yeah.
Do we know what the normal temperature of the brain is?
I mean, it's the temperature of the blood, I guess.
Don't know. Honest question, I have no idea.
I don't exactly know, but I would assume that it's the same as the blood, like the internal temperature.
I would guess so, but as a guess, has anyone ever measured?
Kind of.
We're gonna have fun for the rest of the day. Patrick, you had another question.
So I think the one thing that makes it really weird when it comes to yawning is that we see it in different species. So
so many different species even.
And I don't know, I mean, this cooling hypothesis sounds crazy to me. It sounds kind of outlandish, but hey, I'm not an expert in that. So, but still, it's like we need to have some hypothesis in order to test something. And why do we keep having yawns in modern and concurrent species? So I think that's the strongest argument for that it should have a function or should have some meaning, right?
Yeah. And I think the thermolytic hypothesis is the most kind of obvious one when you think that when you know that it's not oxygen, or at least it doesn't seem to be oxygen. The next one that seems the most obvious, even if it's not super obvious, is the thermolytic hypothesis. But maybe it's something less obvious. I don't know. I have no idea.
I'm just speculating here, obviously.
Yeah, sure.
But it might also be some form of primitive communication that has been, for some reason, stayed within the evolutionary tree. So I mean, the reason why I'm saying that is that we yawn during specific situations, right? We don't yawn randomly, but we yawn if something's boring or if we're tired or something like that. So
I'm blanking on the details here, but I remember reading a study many years ago that you're more likely to mirror the yawn if the person yawning is closer to you.
Yeah, but I think we are an exception because we are a very social animal, and yawn does have a social meaning for us. We are mirroring each other with this kind of thing. So I don't think we can really generalize this kind of behavior to all different species that are yawning.
But there are other social animals, obviously.
Yes.
So it would be interesting to see if that's really a thing, like if the prevalence of yawning is higher in social species versus non-social species.
I wonder if the naked mole rats mirror each other, because they are a very social species and they have a longer yawn than they should. And so that would be interesting to see.
So I just, speaking of social species, I just read a paper this morning about whales and the social abilities in orcas. But like you have to be an animal living in air to yawn, right?
Not necessarily, I think. No, hey, do fish yawn?
Sounds risky.
I don't know. I don't know. I think maybe. I mean, I have no idea. That would be
Yeah,
funny.
you had another question.
Oh yes, I was. I had a comment. I was thinking, like, abandon the idea of oxygen and brain. I was thinking that actually I heard about that when you sleep, your temperature cools down. No, if I'm not right, there are studies. There is a branch like in medicine that study this, and like when, if I'm not wrong, when you sleep, your temperature cools down. So it makes sense, this cooling of the brain, because you know you what's the name? You zone a lot like before sleeping. Maybe it makes sense. I don't know.
But your core
I was just
temperature
thinking.
doesn't drop when you sleep. Your core temperature doesn't drop when you sleep, does it?
It's a little bit like it's lower. There's not a drop, so, but it's lower.
I think so, because the morning it's slower than the afternoon or the evening. I think there is studies showing that.
I don't know. I have no idea.
Yeah, your body temperature reduces when you sleep.
Yes, I remember there were like scientists that studied that in a department in my university. I remember the studies. So I was correlating these two things.
This is truly exciting science. We're starting from one thing and we end with something completely different with thousands of questions that we need to investigate for the rest of the day, or maybe even years to come.
Yeah, that's cool. Let's.
In the interest of time, I would move us on to the next paper, but thank you very much. Very fun start to this week. Hervé, you also had a paper for us, right?
Yes, and it's surprising because it will be about brain size again. So I don't know. We didn't discuss it with Victor, but okay. So
great minds think alike.
So I'm gonna send the link. So so you can see it. Yes. So it's about the evolution of mammalian brain size. So it's strange. We just discussed about brain size. So this paper is about brain size. So why size matter. So it's the relation about between brain and body. So it's talking about the brain-body allometry. So I picked this paper for different reasons, but one reason, so as Victor showed us, brain size is important. So you need to take into account this brain size, and even when you yawn. One thing was interesting is when you study structural brain, so when you do neuroimaging, structural neuroimaging, one main confounding variable you want to take into account is the brain size. The second reason is, so brain size is important for evolution. So there is a lot of stories saying that when you have a bigger brain, you have bigger cognitive capacities. And so that's why I picked this paper. Some example I can give you, if I do that, okay, so this is, okay. So another thing is that there is a relation when you look at brain size, there is a lot of correlation with other factors. For example, if you look at the gyrification of the brain, there is a clear relationship between the gyrification of the brain and the brain size. So that was the interest of the paper. So to come back to brain body size allometry, it went back a long time ago. So this is the study of Dubois in 1897, where they started to plot. So you can see the size of the where is the body? I think the body is here. let's see. So you have the body, I think somewhere. So the brain is here and the body here. So they started to plot these two variables, and so they saw that there was a relation between these two measures. And so here you can see the different species. You have the tapir, the elephant, all the mammals, and also other species. So for the mammals, it's assumed to be, and across different orders, it's assumed to be stable. For example, for the mammal, it's close to 0.5. So what is interesting is that let me change that. Okay. So what is interesting when you look at this curve, there is two things. So when you plot a regression in general, you have the intercept here and you have the slope. So this paper will look at these two factors, which is the intercept and the slope. And more interesting is looking at during evolution if there is a change of intercept or a change of slope. So why they are interested in that? So they want to know, it's stated at the beginning of the paper, so when you have a shift of intercepts, it's meaning, there is a stabilizing selection, so there is a restrained variation. And when you have a shift in slope, that means you have divergent selection. So when you have a shift in slope, it means you have more variations. It allows more variation. And so they wanted to look at that and also to look at the timing of these different shifts. Okay, so what they did in this paper, they look at the, so they took different species, so mammalian species. There is another paper about birds. As we saw just before with Victor, birds are also interesting. But this paper was only about mammalians. So they took 107 extinct species and 1,300 living species. And they started to plot these body and size and estimate. So I will not go to the method. They have a method to estimate properly these shift and in slope and in intercept, but even that, yeah, I didn't get all the methodology behind this paper, but I took the main messages. So the main figure of this paper is the following. So this is a pretty cool figure, where what they did, so based on the slope and the intercepts, they derive different grades. So they divided different groups of species based on the slope and the intercept, so based on the shift of intercept and slope. So just to go back to this figure, so you have in gray, you have the ancestral species, and you can see they derive different groups of species based on the shift on slope and intercept. So to go back to what is on this color, so there is two cuts. The warmer and the colder colors are based on the slope. So for example, if I take the red species, that means there is a higher slope on the allometry between body and brain size. And if you look at the blue species, they have the inverse relationship. So there is a decrease of the allometry. And the other cut of this figure is the darker and light and dark colors. And that means higher or lower intercept. So for example, if I take the bat, compared to other species, the bat has, these kind of bats has a higher intercept. And you can see the hominins here as with both high slope and high intercept. Okay, so on that they derive 30 grades, including the ancestral grade. Okay, so they have 30 grades based on shift of intercept and slope. I hope you are not completely lost now because it took me time to get completely this paper. So it's pretty simple. In fact, it's just body and brain size, but to understand the shift in slope and intercept, it was not so easy. So after they discussed about these results. So first discussion was so they wanted to get a timing of the shift. So they have a lot of shift in slope and intercept. In fact, they have 19. So 19 grades are due to shift in slopes. And what they said, or what they showed, is that this shift. A lot of these shifts are very early. So they date this. They have a datation of this shift. So a lot of them happened 60 million years ago. And why this period? Because we have this big asteroid coming up, and so that these changed the environment a lot, and the selective pressure was kind of important. So it's why we have this shift. And another period where they got some shift was late. It was around 30 million year ago, and it happened for the primates. So that correspond to, I will come back to that later, but locomotion and stuff like that. Okay, so after timing, they started to look at the top big brain. So they look at the species who got the highest in the allometric space. So you have, for example, so they derive or they explain different not strategies but different scenarios. So the elephant is the most simple because it's it got there just by increasing the body and increasing more the brain. So it got here at the top of the allometric space. The other scenario is for the whales. We talked about just before, the whales and dolphins. What happened for them is a lot of intercept shifts. You see the slopes are kind of similar, but just there is a step. It's composed of different steps for the whales and dolphins. And the last scenario, and not the less interesting, is for the primates, which is the most complex, because you have very different kind of thing happening. For example, the hominins, you have so if you take the great apes and the hominins, you have kind of same intercept, but the slope is compared to the ancestral slope. The hominins have a bigger, higher slope, and the great apes have a lower slope. So completely different kind of scenario. So more complex. Okay, and one interesting is that only in these different species, only the hominins, different groups of species, only the hominins and dolphins increase brain size and decrease body size. So often it's an increase of both, and it's a relative speed of both of them that create the shift. But for humans and dolphins, you have the opposite situation. Okay, so after that, they wanted to talk. So here's what I said at the beginning. So when you have shift of slope, they wanted to make these. There is a story about shift in slope and variation and selective pressure. So these three elements. So variance, shift in slope, and selective pressure. So these three elements go together. So they wanted to test, not to test, but to look at the variance, and more specifically, they wanted to distinguish the variance in body and brain size, and look at the shift in slope. So what they, so one sentence I remember is they wanted to demonstrate how changes in the cross-species variance of trait between groups affect shift in slope and may reveal possible selective pressure. So here again, these three elements: variance, shift in slope, and selective pressure. And so you have different scenarios. So here you have the scenario A, where you don't have any kind of change of variance. So you have the same variance for body size and same variance for brain size. What is happening is only a shift of the slope or of the curve. But in this case, there is no so much selective pressure. So here you have two different scenarios, and these two different scenarios are happening in the primates. So for example, there is this kind of variation of variance of body size here. So you have a shift in slope due to variance of body size, okay? And that is happening in the case of the primate where there is a change of locomotion between arboreal and terrestrial. So there is a change of body size which create a change of allometry and a change of slope, okay? Another case is the case C, where you have a variation of brain size and not body size, which create also a change of slope. And that is happening in the hominins, where you have a change of locomotion. And when you go to use your two feet and you go to bipedality, okay? So they make this story about these different change of slope, variation, variance, and selective pressure. The last thing they discussed was cognition, because at the beginning they say, yeah, we are looking at the brain size because we are really interested in, because we know it's affecting the way cognition is done. So we often say bigger brain, bigger eye cognition. As I stated before, one of the main factor of this change of brain-body allometry was not so much cognition, but locomotion. And the other thing is, it's often said that big brain is associated with big associative regions. And they took this example of the polar bear and the sea lions, where the polar bear has a bigger brain for the same body size. So if you look at the allometric measure, that will say that we could interpret that as a bear has a higher cognition level. But when you look at the brain of these two different species, and if you look at the sea lion has a smaller brain, but more regions for the higher cognition. So and the reason for that is because it's using a lot of vocal learning and other cognitive skills. And one of the conclusions of this paper was so when you look, so they try to say, looking at brain body size is important, but taking the brain as a whole is not enough. So we need to go to a level of brain regions and to go back to functions. I want to change the slide, but it's not. Yeah, we just need to go to the level of brain regions to look at the allometry between size and body size and brain size, regional brain size. So that was one of the main conclusion of this paper. It's good because it's what we are doing in the lab. So we don't look at the brain as a whole. We do, but we go to the regions and we look at the function of these different regions and how they interact each other. And that's all.
Thank you very much. Nice fit with the first paper indeed. Are there questions? There was one question in the chat for you, Eri, where the slides can be found.
the slide.
Yeah.
It's strange because in the website they give the paper, but they give you also the presentation. Not all the slides. So if you want to have some specific slide, but yeah, the main slide of the paper are, yeah, you can download the PowerPoint and the PDF.
Okay. Are there any other questions? No. In that case, I have a question about the allometric figure that you nicely explained in the beginning and made the point that it took you a while to get it. And I think I'm not quite there yet.
This one?
Yeah.
Yeah.
It's beautiful, but I kind of got confused when we were talking about the heat gradient in the colors.
Yeah.
And basically the colder and the warmer the color, the different meaning. So you pointed to the example of red being one end of the spectrum and blue being the other end of the spectrum. But then when we look kind of at the bottom of the figure, we see that monkeys, like great apes, monkeys, and humans are all together with dark colors, but completely opposing, but they're quite...
Yeah, so maybe it's clearer here. So here you can see, so it's always, maybe I didn't mention it. It's comparing, it's comparing the it's comparing the intercept and slope to the ancestral slope. So the mammal in general have a slope of 0.56 or 51, and all the comparison are here. So for example, you can see the primates have an intercept higher than the global intercept. So it's why they have a darker color. Okay. So, and in the opposite, if you look at this kind of bat and mice and rat, they are below. So the intercept is below. So they have lighter color. And after, if we go back to the primates, after the color is based on the slope, as I said, the hominins have higher slope as compared to the ancestral species, and the great apes they have a lower slope. It's why it's the color is blue. It's colder.
Okay, cool. Thanks for explaining it again.
Yeah, but it's been a long time. And all the story about the variance was kind of wiped out. I know there is variance, but they do a clear link between variance and slope, which is for me was in terms of statistics, it's not. Yeah, it's not so easy to get it in.
Great. Well, I would leave it up to you now. In theory, we're out of time, but we started late today. So I'd be happy to do the final paper that we have for today or move it to next week. Depends if you guys have more time. And it also depends on you, Valentina, if you want to do it this week or next week. But bear in mind, next week I can't be here. So you wanted to host it as well.
Oh, let's move it to next week. So I can be the host and the presenter.
All in one. Proper multitasking. Fantastic. Okay, in that case, we come to the end of it. Thank you so much for those exciting papers this week. We definitely have enough food for thought for the rest of the week for all of us. Thank you so much. As I said, next week I can't be here, but I'm going to be handing over to Valentina, who's going to do it all. And I hope you have an exciting week in science. See you soon. Bye.
Bye.